not supposed
Oh, really?
to. Yeah. Do you want me to announce it?
Everybody's welcome to come. It's fun.
Rethinking Anomalies in Science is a workshop, but you probably, unless you get here early, you won't have a chair. Because it's very popular. Anyway, if you're interested in attending, you can register online. And then the Tuesday following, on the 31st, a scary day to get a talk, will be Lev Weidman talking about the transfer of quantum information and teleportation, quite an appropriate Halloween topic. There is a call for papers deadline on November 15th for the conference that Arnaud Levy is organizing, Revitalizing Science and Values. So if you know people who might be interested in participating, let them know. They should get their applications in by then. And also, if you know people who might be interested in being a postdoc here next year or a visiting fellow, you should also get them working on their applications for that. All right. And it is my pleasure to introduce Saira Khan, who has a PhD from LPS, one of our sister departments at the University of Irvine, who works on social epistemology, game theory, all those kinds of theories, and feminist philosophy, and is currently a postdoc here at the center, which most of you. So I'm not going to take up more of the time so that we can listen to Saira, and she's going to talk to us about coarse-grained theories of cooperation and Myxobacteria, very famous one.
Thank you, Sandy. Okay. Coarse-grained theories of cooperation and coarse-grained photo Myxobacteria to deal with it. So here's the thesis today. So I'll be talking about a particular strain of Myxobacteria known as Myxococcus xanthus. And this has received particular attention for its cooperative interaction in a realm of different areas in its developmental life cycle, in quorum-sensing, in fruit and body formation, in social motility, and famously predation, which I'll be talking about. So here I'm asking, can our current major theories of the evolution of cooperation account for M. xanthus cooperative predation? As in, can we explain it? So this I'll answer no. I offer two reasons. One is a potential mischaracterisation of the cooperative phenomenon at play as a linear public goods game, or a game in which strategies are operating independently of strategies are independent of genes operating in other M. xanthus activities. Another reason is the insensitivity of these coarse-grained explanatory models to ecological and genomic contexts. So here's the outline of the talk. First, I'll talk about the problem of the evolution of cooperation, which is famous in biology. Then I'll talk about existing explanations of M. xanthus cooperation in other areas of its developmental life cycle. Then I'll explain why these explanations don't apply to explaining M. xanthus cooperative predation, and we'll conclude with lessons learned. So the problem of cooperation. First, what am I calling cooperation? So here is Hamilton's schema for defining social behaviors. Whether a behavior is beneficial or costly depends on the lifetime fitness consequences of the behavior and its absolute fitness effect. I'm going to follow the work of some others before me in referring to cooperation as anything that benefits the other, but may be beneficial or costly to the individual involved. So it'll encompass cases of both mutual benefit and altruism. So here's the problem. Organisms cooperate widely, and we know that. Pack predation is a particularly important form of cooperation. How do I sorry. How do I Thank you. organisms cooperate widely. Pack predation is a particularly studied form of cooperation in lions, animals, so, wolves, humans, and cooperation can lead to synergies which could not have been achieved by actors in isolation. Examples are right. So a group of lionesses can shepherd a deer away from, or whatever they're hunting, shepherd it away from their herd, whereas a single lioness alone might not be able to do so. Yet in many cases, despite being beneficial, cooperation is often risky. So if profit is differentially distributed or involves costly mistakes, some agents may do better by not cooperating or choose to avoid risks, even if they would. Need cooperation is puzzling from a biological perspective. Natural selection should favor those traits which contribute to reproductive success, yet cooperation can be costly to one's own interests. So what would motivate an agent to behave in such a way? How do we explain when an organism engages in an activity which is apparently costly to itself, but benefits another or the wider group? So now let's look at the cooperative phenomenon that we're trying to explain. So Myxobacteria or soil bacterium, they're virtually ubiquitous. They have two types of motility. One is called amotility or adventurous motility, and it's observed for single cells. The other is called S-motility or social motility, and it works in this way. So myxobacterial cells are rod-shaped, and their movement is characterized by gliding in the direction of their long axis. It's characterized by periodic reversals of direction, and movement leaves behind a trail of slime which other cells preferentially travel along, giving rise to population of level patterns of movement. Migration of the other cells through these trails is kind of analogous to the way that we see ants moving in a trail used on the social organization here as untenable chemotherapy systems. The second way in which M. xanthus cooperate is in fruiting body formation. So M. xanthus exists as single cells or small groups of cells during vegetative growth, but in response to starvation, they transition to obligate multicellular fruiting bodies. In fruiting body formation, tasks are divided among subpopulations of the colony, so some transform into spherical spores with the power to withstand desiccation and various temperature levels. Some differentiate into peripheral rods that then move around scouting for food. And some have the fate of programmed cell death, which provides nutrients in assisting the morphogenesis of other cells into spores. So the division of labor here is a cooperative phenomenon akin to eusocial insects again, in that there's different roles played by different organisms, which enables a group benefit. The third way in which M. xanthus cooperate is in predation. Predators have been identified in all three of the myxobacterial suborders, and the predatory strategy exemplified by Myxobacteria is communal. So it involves the secretion of predatory material into the extracellular environment, where this predatory material takes the form of hydrolytic enzymes, such as proteases and lysozymes, into the extracellular environment, which then kill and lyse prey cells and release nutrients. Arguably, all of these activities are preceded by a fourth form of cooperation, quorum-sensing. Quorum-sensing is a term that refers to the communication between bacterial cells to achieve a group-level response. The response is dependent on communication between the cells in the form of production of some diffusible molecules. So in order to orchestrate quorum-requiring behaviors, bacteria signal to each other that they are present through the secretion of these chemicals. Here's a very stylized picture of their life cycle, where the things on the slightly blue side are what happens in nutrient-rich environments, and the things on the white side are what happens in nutrient-sparse environments. So how is M. xanthus predation cooperative? It's cooperative in at least two ways. First, predation requires a quorum. So it involves a minimum number of attaching cells to lice prey. Second, predation also relies on social motility, which we know to be, it relies on motility, which we know to be at least social. Indeed, predatory activity was completely abolished in mutants lacking both motility systems. There's another way in which motility matters for predation. So arguably, regulated reversals are essential for the characteristic rippling pattern of xanthus during predation. And the idea is that they synchronize their motility and reverse in oscillating waves. And it's been suggested that they produce a signal that allows them to respond to head-to-head collisions with other xanthus cells during this behavior. Rippling in the presence of prey increases the rate of colony expansion by the predator and reduces the mean square displacement of individual cells, which allow the predator cells to remain within the prey area longer. So some have argued that rippling is not completely a necessary part of predation, but we know that some kind of motility is at least required for the searching of prey. I'll also mention that in some cases they do individually hunt as well. So the extent to which this is a cooperative phenomenon is still being debated and still being tested in experimental studies. But we know at least in some cases it is cooperative. Okay. So here's a video. I don't know if it'll work. Okay, so this is what the predation looks like. Here we're going to see xanthus gliding across a surface in a socially coordinated swarm. And when they make contact with the prey, this is an E. coli colony, then the rippling behavior occurs, and cells that leave the E. coli colony will resume normal colonizing behavior or aggregate into fruiting bodies. So the task is to explain the stability of these cooperative practices in the face of actors who don't contribute to who don't contribute to the cooperative activity and instead free ride. So there are a few examples of how this might happen, the problem of cheating. So in quorum-sensing, a cheater might not produce the quorum-sensing signal, and it therefore benefits from the efforts of other cells in producing this costly signal and monitoring local cell density without itself undergoing cost associated with signal production. Alternatively, a cheater might overproduce the signal but not respond to it. So this can result in neighboring cells producing more of the public good, while the actor who doesn't produce it, who overproduce it doesn't produce it. As I mentioned earlier, the division of labor as well is akin to social insects with the division of responsibilities among different agents. And so a cheater here might not perform their role, but benefit from the increased sporulation efficiency of the fruiting body. So according to findings by Balser and Viegna-Balser, if cheaters in multicellular development persist to represent a large percentage of the population, the ability to undergo cooperative interactions and purposes dramatically decreases. Indeed, outright extinction was reported in some laboratory studies after the introduction of cheating genotypes in fruiting body formation. In predation, cooperation is costly to the individual in the production of enzymes and secondary metabolites. And the question is, how do we explain the stability of cooperative behavior in the face of potential exploitation? The form of exploitation that this would take is that free riders could underproduce the predatory material and still benefit from the subsequent release of prey nutrients by others who do so. So we're going to talk a little bit about game theory just to make this problem more precise, and then we'll look at the proposed solutions. So I'm sure you'll all be familiar with the prisoner's dilemma game, and it's often invoked in explaining the evolution of cooperation. The options are to cooperate or defect, and defection is the Nash equilibrium. In cooperative predation, we actually have a public goods game. So this involves multiple agents, not just two. And in the public goods game, the agents have the option of giving some of their endowment to a public pool, which is then multiplied and then split among the agents. So the group's total payoff is maximized if everyone contributes all of their endowment. However, the Nash equilibrium is to contribute zero. And that's because if you're contributing zero, you have no unilateral incentive to deviate from that strategy. So the best thing here to do is to let everybody else contribute and gain from their contributions, but do nothing yourself, so to free ride. In the case of myxobacterial hunting and other animal hunting, arguably we're faced with the public goods game. So many theories have been offered of the evolution of cooperation, and here are some of them on the side. I will cover all of the theories of the evolution of cooperation, but I'll cover the ones that are salient to our current interests. In particular, we're going to exclude theories of the evolution of cooperation that rely on reputational benefits, since myxobacteria don't have reputations, so we won't be talking about indirect reciprocity or network reciprocity. And we're also going to exclude things that rely on cultural evolution, since myxobacteria don't have culture. So here's one explanation of the evolution of cooperation, kin selection. Here, cooperation is selected for, in simple terms, if it has a positive inclusive fitness gain for others related to the individual, or those who bear the relevantly similar cooperative gene. So there's sort of two forms of kin selection. One is about relatedness. So I might sacrifice my life on the Hamilton's rule if it saves two of my brothers, because each of them have half of my genetic material. There's a broader sense of kin selection which applies to cooperation outside of just those ones who are genetically related to you. So this is known as the green bit effect. If someone bears the same cooperative gene, whatever that is, and also has a phenotypic marker that's associated with that gene, then if we preferentially direct cooperation towards those with those phenotypic markers, we'll be preferentially directing cooperation to other cooperators and proliferating the cooperative gene. Another explanation proposed is signaling. Simply, cooperation is selected for if it is preferentially directed to other cooperators via pre-place signaling. So the signal allows cooperators to identify other cooperators, and therefore they're going to avoid exploitation by defectors because they don't interact with them. Another form of ensuring cooperation is punishment. Most simply, punishment is just punishment for defection. So defection entails a fitness cost, which means that cooperation is preferential. A few more. Reciprocal altruism. So here, cooperation is selected for if it directly benefits the cooperator, usually through some sort of like treatment in kind. So individuals pay a current cost for cooperation in return for a social partner's reciprocation in the future. An example is a cleaner fish who cleans the mouth of larger fish who then refrain from eating it. So both benefit from this form of cooperation. Another explanation of cooperative behavior is commitment, where cooperation is selected for if it benefits the long-term fitness of the cooperator by constraining short-term incentives to cheat. So for Schelling, who introduced commitments, these are strategic moves which induce another player to choose in your favor by altering your own incentive structure. So if I tear off my wheel in a game of chicken, you can see that I've changed my incentive structure. You're incentivized to swerve, I drive forward, and I win the game of chicken. But you could equally well apply that to a cooperative situation. So the chicken game is a zero-sum game. You could equally well apply that to a cooperative game like a stag hunt. And finally, group selection. So here, cooperation is selected for if cooperative groups outcompete non-cooperative groups. So Sober and Wilson's version of group selection argues that altruism can evolve or cooperation can evolve as different groups make different contributions to the same reproductive pool in which new groups are formed. And if a group with a higher proportion of altruists has greater reproductive output, it can be shown that the resulting proportion of altruists is greater than that of both initial groups combined. So now we can see whether any of these explanations account for xanthus cooperation. In particular, first I'll go through the explanations that have been proposed already to account for cooperation in other areas of the xanthus life cycle. So in their quorum-sensing, fruiting body formation, and social motility. So kin selection. This has been proposed to account for quorum-sensing. As we've just discussed, quorum-sensing molecules act as a proxy for cell density, so they precede a density-dependent cooperative behavior. And we've seen a couple of ways in which you might cheat by either overproducing the signal or not producing the signal. Brown and Johnstone in 2001 developed a kin selection model of quorum-sensing. So they examined the consequences of variation in mean population density and relatedness. And they found that the evolutionarily stable strategy for signaling and public goods production both increased with greater population densities and greater relatedness. One mechanism suggested to overcome kin dispersal and facilitate kin selection in the case of fruiting body formation is what's known as the outer membrane exchange. So the outer membrane exchange is a bidirectional transfer of outer membrane components, including lipids, lipoproteins, and lipopolysaccharides between two bacterial cells. And the way in which this is supposed to work is that there are two surface proteins, Ture A and Ture B, and they act like Velcro hooks to bring cells close enough for OME to occur, but they also facilitate recognition. In particular, Ture A proteins only recognize identical forms of those on the other bacteria, so this prevents M. xanthus from accidentally helping distantly related M. xanthus populations. Signaling is another method of maintaining the stability of cooperation in the face of the potential to cheat. And this has been proposed to account for motility. So to explain how cooperation is maintained in social motility, Ducret et al. have suggested that slime-embedded outer membrane materials or outer membrane vesicles could contain signals that would promote specific recognition and facilitate trail following. It's also been proposed to account for fruiting body formation. So in fruiting body formation, coordinated cell aggregation into multicellular fruiting bodies is regulated by cell-to-cell signaling. During initial stages of development, M. xanthus cells accumulate elevated levels of what's known as PPGPP in response to starvation. And this generates what's known as an A signal. The A signal is an extracellular quorum-like signal that's thought to monitor local cell density, and once this achieves a minimum threshold, the expression of development-specific genes related to fruiting body formation are initiated. Finally, punishment is also proposed to account for fruiting body formation and motility. So in fruiting body formation, it's been found that in the laboratory, Myxobacteria can evolve policing capacities. They control cheating associated with a CSGA mutant, where the CSGA gene is responsible for cell-to-cell signaling and development. In motility, cow-et-al, though this isn't in service of cooperation, it's actually in service of not cooperation, cow-et-al find that motile M. xanthus cells are inhibited from swarming when mixed with the non-motile cells. And they concluded that the non-motile cells are killing the motile cells by expressing a toxin-antitoxin pair presumed to be delivered by a TRA-dependent OME mechanism. So here's a summary of the proposed explanations of cooperation in other areas of the M. xanthus life cycle. So in fruiting body formation, quorum-sensing, and motility, we have that kin selection has been offered as an explanation for quorum-sensing and fruiting body formation. Signaling has been offered as an explanation for motility and fruiting body formation. And punishment has been offered as an explanation for fruiting body formation and possibly quorum-sensing. So I don't think that these explanations account for M. xanthus cooperative predation. In particular, the problem that we're trying to explain is why, how we overcome the incentive to free ride by not producing or underproducing predatory materials, hydrolytic enzymes and secondary metabolites, and just benefiting of the free riding, just benefiting of the contributions of others in the release of prey nutrients. So I'll explain why I don't think the same mechanisms account for predation. So first, kin selection can't offer an adequate explanation since the major mechanism proposed to account for kin selection, it is through OME. And here the goods are exchanged in a contact-dependent manner. So an advantage of cooperation fruiting body formation is if the goods are exchanged only after the involved parties are verified in a cell contact-dependent manner, we can ensure cooperation is achieved. This is true of predation. So in predation, secreted public goods are poorly controlled once they leave the cell boundary, and bystanders can take advantage of this. So remember, the predatory materials are released into the extracellular environment, and this is where the prey cells are killed and where the nutrients are released. So there's no contact-dependent manner in which we can ensure the fruits of cooperation are protected among cooperators. I don't think signaling will suffice, as it's possible that xanthus predators are not signaling at all during predation. Some people have argued that xanthus don't make quorum-sensing signals, they don't make AHLs themselves, but rather these are produced by the prey organisms, and it stimulates predatory activity. So some people have referred to this as eavesdropping on prey conversations. So rather than the predators cooperating and signaling to each other that the prey is present, or signaling in order to achieve to initiate quorum requiring behavior, they're just individually responding to the presence of prey. Finally, I don't think punishment accounts for xanthus predation because there's just no evidence of punishment for cheating in xanthus predatory activities. But more than this, I don't think other theories that I've given you can account for xanthus cooperative predation either. So I talked about reciprocal altruism, commitment, and group selection. So I'll go through those in turn. In the case of reciprocal altruism, there's no dividing of the good in order for reciprocal exchange to occur. And of course, there's no indirect reciprocity that's dependent on a reputational mechanism. So another way of putting this is that reciprocal altruism is a good explanation of the evolution of cooperation in dyadic interactions over time. So I help you now, you help me later. But it's not a good explanation of the evolution of cooperation in simultaneous public goods games. In the case of commitment, this requires a pre-play signal which introduces a constant affection, and arguably we don't see either of these in the case of Myxobacteria. So I just told you that in the case of signaling, individual cells might not be signaling to each other before engaging in cooperative predation, rather they're just individually responding to the presence of prey. And in the case of the cost of defection, this requires some sort of punishment for not following through on one's commitment signal. And we don't see punishment, or at least we haven't seen punishment evidenced in Myxobacteria. In the case of group selection, so the first thing to say is that in vitro experiments, Myxobacteria colonies are spatially isolate. There's no between group selection. So what we need is a natural study to see whether group selection actually holds in the wild. In particular, we would need to show that xanthus populations were subdivided into competing groups over their evolutionary history, and that there are mechanisms which sustain between group variation and mechanisms which affect a group's proliferation. So this remains a live possibility that group selection is an explanation for the evolution of or the stability of the cooperative predation of xanthus, but it's as yet insufficiently tested. And there's reason to suspect that it's maybe different in the cooperative predation case than it is in fruiting body formation, because in fruiting body formation, there's a group level property with fitness consequences. So altruistic, as I said, a certain proportion of the cells have the fate of programmed cell death, and altruism here releases nutrients to assist others into other cells into the morphogenesis and spores. But there's no, and this increases the sporulation efficiency of the whole group. So it's altruism, and there's some group level property that's increased, that has increased fitness. But there's no such analogy in the case of predation. So we would need more evidence basically to assess if group selection was a viable explanation of cooperative predation in xanthus. So I've considered whether our major theories of the evolution of cooperation can adequately account for cooperative predation. And in answer to whether they can, I've said no. And in what follows, I'm going to give you 2 reasons for why I don't think that they account for it. So one reason we may not have come to an adequate explanation of cooperative predation in M. xanthus is we may have mischaracterized the cooperative game under consideration. So it's frequently presumed that M. xanthus cooperative predation is well represented by a public goods game, a traditional public goods game, which is linear. So multiple actors have the option of contributing to the same public pool. The sum is then multiplied and divided among them. But what if this isn't the thing? So in a linear public goods game, we know that some form of the Nash equilibrium is 0 contributions. So we know some form of positive assortment or enforcement is needed to ensure that cooperation is maintained. But perhaps M. xanthus cooperative predation is better represented by a non-linear public goods game or a threshold public goods game. Indeed, people like Archetti argue that biological public goods should be modeled using sigmoid or step functions rather than linear or concave functions. I'll show you a photo of what these mean in a second. And some cases that they give to support that conclusion are that in the case of diffusible molecules, non-linearities arise because of the effect of biological molecules on cell fitness. It's typically a sigmoid function because of their concentration due to threshold effects or due to cooperative binding of ligands. Or an easier case to understand, in social interactions, non-linear linearities arise when just a portion of the group is large enough to detect predators or take down a prey. So the kind of situation you get is something like this, where so a linear function obviously would just be a straight line. But you could have a piecewise linear function, a sigmoid function, which is the S shape, or a step function, which increases. So the step function would represent a threshold public goods game, where some level of contributions is needed for the public good to be realized. So for a minimum level. And a sigmoid function is where it's not linear, basically. So the important thing to note is that if we have a sigmoid function, cooperators and defectors can stably coexist in an equilibrium when the cost-benefit ratio of cooperation is below some critical value. So we don't need punishment, signaling, or kin selection as an explanation for cooperation in this case. Cooperation is maintained endogenously in the model. It can be shown mathematically, we don't need to go through all the math, but that higher inflection point in the sigmoid function means that there can be a higher stable proportion of cooperators, and the steepness of the curve affects the ability to achieve equilibrium at high cost of cooperation. So here the explanation is just that the explanation of the stability of cooperation is just direct benefit to the cooperator. So if non-linearity is a true characterization of M. xanthus cooperative predation, then we don't need kin selection, signaling, or punishment to explain it. And the same goes for a step function, which would represent a threshold public goods game. So in a threshold public goods game, say you need K cooperators for the public good to be realized. If you're the K minus 1 cooperator, it's in your individual benefit to cooperate to get the public good to be realized. So free riding only arises if there's redundancy, like more people than is necessary to achieve the public good. So here, cooperation is sustained at the group level simply because it's required for any benefit to be received by the individual actor. Another way in which we might have mischaracterized the game is by considering selection only operating on strategies, individual strategies, rather than on genes which influence multiple strategies. So we know the predatory ability of M. xanthus is negatively affected by mutations implicated in early stages of development, so mutations in the following genes, as well as genes of the chemotactic system, which modulates motility and development. So previously, we've considered all their cooperative interactions separately. So we've considered fruiting body formation, social motility and predation as happening as being represented by separate games where selection is operating on individual strategies within those games. However, it's possible that it's better modeled by some sort of cross-cut game and this would change our analysis of the explanation. So if selection is operating on genes which are implicated in later cooperative activities, then cooperation in other areas of the developmental cycle will have implications for downstream cooperation in future areas. So the free rider problem might not arise even in predation if, for example, gene expression of the production of predatory materials was systematically linked, say, to the development of breeding body formation, which is poorly explained by other mechanisms. Okay, a second way in which I think that we've gone wrong in trying to explain cooperative predation in xanthus is being insensitive to ecological and genomic contexts. And so here I'm drawing on the work of Karina Tarnita, who argues about the importance of ecology in understanding social behavior in microbes. And she argues in particular that focus on cooperative models, or what I'm calling in general these coarse-grained theories of cooperation, can come at the expense of consideration of ecological context, alternative hypotheses that might reveal different roles and different types of interactions between the purported cooperators and cheaters, depending on genotype and environment. And that's a very dense sentence, so I'll give you an example of what that means. And the possibility that cooperation or cheating are not actually ends in themselves, but represent a transient phase. So the first one first, in support of this first point, I mean, the first thing you want to say is that ecological context is missing in laboratory studies. So evidence has pointed to ecology as being a major determinant of the kind of cooperative interaction that animals engage in and emerging group formations that they have and their social organization. For example, spatiotemporal environmental variation and breeding habitat is an important determinant of cooperatively breeding birds. The dispersion and availability of food and its typical pattern of predator avoidance in African antelopes is a crucial part of its determining its group size and social organization. And so the idea is we just need to pay heed to ecological context when understanding cooperative interaction. And in vitro studies of xanthus couldn't possibly capture this. Indeed, in the bacterial case, it's been found that bacteria behave differently in competitive contexts in structured and unstructured environments. So there's a case study using allelopathy as its example. So allelopathy refers to the production of toxic compounds by bacteria, which kill competitors, and the success or failure of this mechanism depends on whether the toxin producers are in a structured or unstructured environment. In unstructured environments, such as a well-mixed liquid culture, toxin producers are eliminated at low frequency. However, in a structured environment, such as an agar plate, cells that produce the toxin have an advantage even at low frequency. And the idea is that the local frequency of toxin producers is higher than their global frequency, and they benefit from local competitive damage. On Tarnita's second point on genotype environment interactions, she doesn't talk about genomic context very well. She cites a case study about pyoverdin, which is produced by bacteria from the genus Pseudomonas. So pyoverdin is an iron-placing molecule. However, predictions about whether pyoverdin can be exploited by cheaters depend strongly on both genotype and environment. So what she intends to show is that bacteria are not simply cooperators or defectors. That's too coarse-grained. There may be sensitivities to the environment and genotype that changes their expression of cooperation or defection. On genomic context, Charlotte et al. Have produced disruptions of the gene CSGA in diverged genomic backgrounds, where genomic background refers to the genotypes of all other genes that may interact with the gene of interest, and therefore affect the specific phenotype. They demonstrated that whether a mutation generates a cheating genotype will depend on its genomic context. So Note that this would also limit the ability of any given cheating mutation to spread by horizontal gene transfer, for example, given different genomic backgrounds. So again, what this is intended to show is that the cooperated vector labeling in our coarse-grained models are not fine-grained enough. In particular, they don't look at genotype environment interactions in genomic contexts. So it would be a mistake to label xanthus cells as cooperators or effectors without consideration of these things, the ways in which the expression of cooperation or affection could be impacted. Finally, on the possibility that cooperation and cheating are not ends in themselves but represent a transient phase in Myxobacteria evolution, there was a study by Viegna et al. That looked at one cheetah which fails to produce viable spores in monoculture. In laboratory experiments, this cheetah eventually mutated into a novel social type. Descendants of the cheetah strain re-evolved the ability to sporulate independently and overtook the previous cooperative population in mixed culture, also demonstrating additional advantages such as growth in a liquid medium. This suggests again that the characterization of our cooperative models are not fine-grained enough. The evolution of new strategies might be rapid enough that they couldn't be excluded from our models. And finally, above the two reasons, mischaracterization of the game at hand and also ecological and genomic context for thinking that our theories of the evolution of cooperation are too coarse-grained to capture Myxobacteria cooperation. Then there's also a kind of third interconnected reason. So it's important to know that ecological context may also play a role in mischaracterization of the game at hand. So not only may the environment or gene-environment interactions change an organism's expression of cooperation or affection, but they can change the game being played as well. As an illustrative example, consider the hunting strategy of a pack of animals in an environment which is heavily wooded versus the hunting strategy of a pack of animals in an environment which is barren. In the former, ambush hunting perhaps may be more likely, and in the latter, persistence hunting. And the number of actors, the strategies, the free rider incentives will all vary with features such as these. So environment's also important in determining which game is being played. Okay. Hopefully you're not as sad as this kid after hearing all of this. But lessons learned. So I'll just summarize what I've argued so far, and then we'll see what the upshots are. So I noted that it's been argued that cooperation in xanthus social motility, brooding body formation, and quorum-sensing are explained by kin selection, signaling, and punishment. I've argued that xanthus cooperative predation cannot be sustained by the same mechanisms. I also argued that it cannot be well explained by other coarse-grained theories of the evolution of cooperation, i.e. reciprocal altruism, commitment, and group selection. I provided two main reasons for our lack of, the inadequacy of these explanations. First, a mischaracterization of the cooperative problem as a linear public goods game, when in fact it might be a non-linear public goods game. And also focusing on selection as operating on strategies in particular individual games rather than on genes operating across multiple games. And finally, the insensitivity of these coarse-grained explanatory models to ecological and genomic context. Okay, here are the upshots. So first, xanthus predation requires greater empirical attention. So we want to know from our studies of xanthus, in order to give an appropriate answer to why cooperation in this sphere is stable, we want to understand under what circumstances they engage in individual versus collective predation, whether signaling always precedes cooperation, whether there is punishment for defection from predatory activity, to what extent disruption of genes involved in other areas of cooperation, such as brooding body formation, cause disruption in predatory capacities, whether there is a threshold level of contribution of effective killing and lysing of prey cells, so whether it's well represented by a threshold public goods game. And how xanthus behave in their natural environment, which will give us clues to group selection. So also we want to know whether the conditions for group selection are met over their evolutionary history. And the answers to all these questions will help our understanding of how to model this cooperative phenomenon precisely, as well as help us to answer the question of how cooperation is sustained. Second, we want a new model to represent predation, and this could lead to new avenues for explaining xanthus cooperation, how it is stable. So, as I've argued, if represented by a non-linear threshold public goods gain, individual benefit might just be explained by, might just explain the stability of cooperation itself. In some sort of composite game, cooperation in predation would depend on cooperation in other areas of the xanthus life cycle. And finally, and most importantly, I believe this analysis provides insight into when and where our simplified cooperative models can provide good explanations of real-world behaviors. So in discussing the importance of ecological context in Myxobacteria, Carnita also appeals to a number of examples in animals, as I've said, the cooperatively breeding birds and African antelopes and a bunch of others. So she also talks about ants as well. And so ecological context is something that's missing from these models and something that means that our explanations are not generalizable. Our theories of the evolution of cooperation in particular are only approximations of mechanisms which sustain cooperation, and they're not generalizable across different contexts. But that's far from saying that these theories of the evolution of cooperation are not useful. In fact, I think they're very useful. They lay the theoretical groundwork for empirical study. So it's only after we've come to theories such as kin selection that we can examine to what extent the conditions for kin selection are met in the real world and explain social behaviors that we see in the real world. But you need caution, you need to exercise caution in not replacing the neatness of the theory for empirical precision when you're explaining a real world behavior. So empirical studies which seek to validate these theories should pay close attention to the things mentioned here, in particular to the characterization of the game at hand, to the natural environment of organisms, to the interaction of genotype and environment, to the genomic context, to the potential for transient phases of cooperation and defection. And perhaps a more integrated approach to explaining cooperative behavior would look to ecological evolutionary developmental biology, or eco-evo-devo bio. So this integrates concepts such as developmental symbiosis, plasticity, niche construction, and things like that.